Hello, welcome back. Continuing on through chapter 7, we are now into the section talking about mutations. We're going to cover the different types of mutations and mutagens, the things that can actually cause mutations in DNA. All right, so again, some of this might be review from your general bio class. I know I do teach this in Bi 211, so it's always good to see it again and reinforce information that you might already have bringing into the class, this prior knowledge. So this type of mutation we're going to take a look at is the effect of a points mutation. So a points mutation, also sometimes called a base substitution, is where you have a single base change in your DNA. And what are the effects on that in the subsequent protein that's coded for by that particular gene? So in the top picture here, we have the normal code. We have the DNA strand, just that one half of it. So the blue or purplish DNA is the template strand. And then the complementary mRNA, right? So A's with U's, C's with G's, T and A. And then the codons. So UUU codes for phenylalanine, UAU codes for tyrosine, GCA codes for alanine, GCU codes for arginine. And remember, that comes from your understanding of the process of translation, which you can again go back and review in those pages we're skipping over with the genetic code and all that kind of stuff. So that's why it's important that you know these things so you can understand the application. So looking at the genetic code, we can see that those codons of the mRNA code for these particular amino acids. So phenylalanine, tyrosine, alanine, and arginine is the normal sequence for this particular gene. But in a point mutation, let's say that third A gets turned into a G in the DNA. So then that's going to have an effect on the RNA. So instead of being UUU, the codon now becomes UUC. And if you look that up in the genetic code, you will find that UUC codes for phenylalanine. Now, if you look at what it was in the normal amino acid sequence, it was also phenylalanine. So a silent mutation has no change in the amino acid sequence. So even though there's a mutation in the DNA, the organism there's no effect on the phenotype. The genotype is changed, the phenotype is the same because it codes for the same amino acid, the protein's going to be just fine. Right? So that's called a silent mutation. If that mutation, that point mutation happens somewhere else, so here it's happening in that G, it gets turned into a C. So normally we had GCA, now we have GGA. So if you go and look at the genetic code, GCA coded for alanine, but GGA codes for glycine. So all of the other amino acids are correct. So we have phenylalanine, tyrosine, and arginine are all of the same amino acids in the correct placement, but we have a different amino acid. And so this is what we call a missense mutation. It's just the wrong amino acid is put in some place along the protein chain. And that could have varying effects, a wide range of effects. It could be very slight, like the protein mostly works, and so the organism may not have any impact, or it could be pretty devastating. So you probably have heard about sickle cell, maybe saw that in your genetics unit. Sickle cell is a pretty devastating disease with the result of a single points mutation at one particular base that changes one particular amino acid at a very important location in that protein chain, which affects how the hemoglobin molecule folds into that three-dimensional shape. And you should also be super comfortable with the idea that a protein's three-dimensional shape dictates its function. That's super important. So if you change that three-dimensional shape, you've changed the function of that protein. All right, so this is what we call a missense mutation. And then lastly, let's change this T here. It was an A, look, A, T. It was an A, and so now it's going to be a T, and so that's going to change that to an A. So instead of having UAU coding for tyrosine, you get a UAA. So looking at the genetic code, this codes for a stop codon. So anytime a points mutation, this base substitution, inadvertently puts in a stop codon somewhere along the length of the protein, you're going to get what's called a nonsense mutation, which stops protein synthesis. And then everything else after that just done, because what happens to the ribosome when a stop codon shows up? The release factor comes in, the large and the small subunit of the ribosome break apart, and your protein's done. In this case, your protein is just phenylalanine. That's not going to do you anything. So nonsense mutations are pretty devastating in the effect of what they can do on the functionality of that protein. All right, so these are all effects of points mutations. Again, that should be reviewed from your general bio. Then we also have frameshift mutations. So these are also pretty darn devastating because what they do is not only affect just one position in that codon, but wherever the frameshift happens, everything downstream to that shift is affected. So we're using the same coding strand, mRNA, and protein chain that we saw in the last one, so phenylalanine, tyrosine, alanine, arginine. And we have two ways that you can do a frameshift mutation. You can have an insertion where the DNA polymerase accidentally puts in an extra base that's not matched up to anything, or you could have a deletion where DNA polymerase accidentally removes a base, and so now there is you're missing part of the DNA sequence. Now, what this does is it shifts the reading frame for the ribosome when it starts bringing in the correct amino acid, what the tRNAs are going to recognize with their anticodons. So if you remember, we had UUU, UAU, GCA, and CGU. Those are the normal codons. All right, so here's our first one, UUU, that's the same. But when we added in this letter, our UAU is right here. But the ribosome's not going to know to skip over that extra A. So what it does is it's just going to read the next three letters, which is AUA, and then the next three letters. And so the whole reading frame has been shifted, and it's going to bring completely different amino acids. And so a major difference in amino acid sequence, this frameshift mutated protein is just going to be gibberish. It's not even going to be functional. And it's going to be the same thing if you did a deletion. So you'd have your UUU, but instead of having UAU, it's UG and CAG, and then, oh, that's not going to do you anything, and so it's going to be done. So again, you have a major change in the amino acid sequence. And so these are also considered points mutations if it's one base insertion or one base deletion. If it's a substitution, it's called a base substitution point mutation, or insertion or deletion point mutation. Any of those can have these wide-ranging effects, from no effect at all with the silent mutations to devastating effects like these frameshift mutations. All right, so those are the different types of mutations. Take a look here. Okay. So now we're going to spend a couple slides on taking a look at mutagens, things that actually cause mutations. So what's going to make a base a frameshift mutation, insertion or deletion or a substitution? So they happen randomly, but there could be also some things in the environment that could increase the likelihood of those things happening. So there's two types of mutagens your book talks about: radiation and chemicals. For the radiation, there's what we call ionizing or non-ionizing radiation. Ionizing radiation is like gamma rays, X-rays, really strong nuclear radiation. That is ionizing because the energy of those, the wavelengths can come in and actually create ions. They can knock electrons out of their atom structure, and those are really devastating. They can actually ruin the molecule of DNA directly and cause mutations and things like that. In non-ionizing radiation, like ultraviolet light is an example of non-ionizing radiation. We have that pictured right here. So just sunlight, right, or a UV lamp. We don't chemically change the atoms, like we're not shooting off electrons out of atoms affecting their reactivity. But in ultraviolet light, this non-ionizing radiation, what does happen is if you have two thymines right next to each other in your DNA strand, it causes them to bind neighbor-like next to each other. And when you have this, it's called a thymine dimer, and it creates a big bubble on this little lesion, if you will, on the DNA. So if you are a DNA polymerase, let's say you're an enzyme coming along and you're reading these bases and you get to this bubble, you're like, what? I don't know what that's going to be. So when it's making its complementary strand, it's just going to do its best guess, which could be the wrong guess, or it could be a deletion or an insertion causing a frameshift mutation. So these thymine dimers mutate DNA that way by putting in the wrong base because they just can't read that template strand to make the proper complementary side. Now, this is what happens to our bodies when we are exposed to too much ultraviolet light, right? So if you get sunburned, repeated sunburn, you are increasing the chances that you are getting those thymine dimers. And so when your cells go to replicate, like your skin cells, they might mutate and you could get carcinomas, right? So basal cell carcinoma, squamous cell carcinoma of the cells of your epidermis. Or you could get malignant melanoma, which is the worst type of skin cancer, and the mutations would be taking place in your melanocytes, those cells that make the pigment that give your skin its color, because of these thymine dimers. So now you know the major cause of skin cancer is mutations in those proteins that code for cell cycle regulation within your skin cells due to these thymine dimers. So long sleeves, sunscreen, right? Protect your skin. Another type of mutagen are chemicals. So we have what are called nucleotide analogs, or these mimics. And so what they're doing, they're chemicals in the environment, or you might be exposed in some condition. So here is our normal structure for thymine. Right? It's got a methyl group right here, the CH3. There's a very similar molecule called 5' bromouracil. And if you'll notice, there's a bromine atom instead of a CH3 methyl group. Everything else in the molecule is exactly the same. So if in your body you are exposed to this 5' bromouracil, your enzymes cannot tell the difference between a thymine and a 5' bromouracil. So it gets incorporated into where a thymine normally would be. Okay, so now we have the 5' bromouracil in your DNA, and then when your cells go to replicate, the RNA or the DNA polymerase is coming along, right? This enzyme reading that and saying, I don't know what that is. I'm going to do my best guess at putting in what that is, because that bromine atom is really big, and so it kind of the rest of the 5' bromouracil looks enough like thymine that it can sneak by, but then in the replication process, it doesn't quite act the same as thymine. So in this case, a guanine was put in place instead of an adenine. And so now we have this permanent mutation within that line of DNA, and that could be your base substitution, which could lead to a silent, a missense, or a nonsense mutation in that particular example. So those are mutagens, radiation examples, and chemical examples. Okay. All right, and so here is another chemical example of a frameshift mutagen. We saw some of the other ones like the thymine dimer could trigger frameshift. This is a very specific type called acridine. And so this really big molecule, and it has a good ability to kind of squeeze itself in between the two DNA backbones. And so again, when you have this big bubble in your molecule, the RNA or the DNA polymerase doesn't know what to do with it. So it could either delete a whole bunch of bases, or it could insert a whole bunch of bases, which is then going to perpetuate in the DNA and then be transcribed and translated with the wrong amino acid sequence. Okay. So that might be a little bit scary for you, knowing that we have these ways that your DNA is going to get mutated, but we do have some repair mechanisms. And I know I'm saying it's the same for both bacteria and eukaryotic cells. So we have similar types of repair mechanisms. So there's two major groups. There's called direct and indirect, or single-stranded, direct repair and single-stranded repair. Of the direct repair mechanisms, we have what's called light repair and base excision repair. So light repair is an enzyme called photolyase, this enzyme here, interestingly activated by visible light, repairs the damage done by ultraviolet light. So what it does is it cruises along the DNA in the presence of visible light. It gets activated, this enzyme, and breaks those thymine dimers and repairs that bond that wasn't supposed to be there. So this is a visible light activated repair enzyme breaking those thymine dimers. In base excision repair, there's another set of enzymes that go along and they try to find, hey, that is not a correct match. And so what they'll do is they remove the incorrect piece. In this case, it's a hypoxanthine, which is probably a mimic of adenine, and it removes that, right, and then fills it back in with the proper base. So this is called base excision repair. It's a direct repair of the damage to that piece of the DNA, either the thymine dimer or the incorrect base being put in. In a collection of repair mechanisms called single-stranded repair, we have what's called nucleotide excision repair and mismatch repair. So to remove those thymine dimers, we could use a photolyase, but we also have another enzyme. They're just generally calling it a repair enzyme, not a super cool name. But instead of just directly breaking those thymine dimers like we saw in the direct repair, they will actually cut either side of the lesion, pull that out, and DNA polymerase will come in and put in the correct bases. And that repairs, it removes that thymine dimer. So this is kind of nucleotide excision because we're cutting out, we're excising the mutation and replacing it with correct bases in this single-stranded repair. And then we have mismatch repair, which is also similar to what we saw in the direct, where they would just remove the one base, base excision repair. This is mismatch repair. So here's a mismatch, but instead of just swapping out the one base, they take out the whole chunk. And then DNA polymerase comes in and puts in the correct bases. So single-strand repair is more of a large-scale removal and replacement, where the direct repair was just you're fixing that one thing. All right, so those are our different types of mutations and how we can fix those mutations. Next, we're going to take a look at how we can identify mutants, bacteria that actually have mutations. All right, so I'll see you next time.